Okay, now we're going to do the second activity. The second activity is isolation. Okay, and what we're going to do is we're going to take two different microorganisms. One is Micrococcus luteus, and the other one is Marcescens red. So this is kind of fun. Micrococcus luteus is a ubiquitous soil organism, and when it grows a pretty yellow color. Serratia marcescens, if you see indwelling cathars, sometimes in the shower, sometimes next to the commode, you see that pinkish color coming out. That's Serratia marcescens. It grows a red color at room temperature. So what we're going to do is we're going to activate Micrococcus luteus and Serratia marcescens, and we're going to purposely mix them together to see if we can do what's called a streak plate, okay? And in a streak plate, what you do is you start out with a plate like this and a nichrome loop like this. And what you do is you streak the plate and then you change the nichrome loop 4 times, so that every time you streak the plate, the concentration of bacteria decreases more and more and more. When you're done, if you're successful at isolation, you should have a nice, pretty red colony of Serratia marcescens separated from a nice yellow colony of Micrococcus luteus. Okay. Also, the streak plate should look pretty, aesthetic. Okay. Everything that we do, you know, if you care about it, it should look nice. So our first job here, of course, is going to be to rehydrate the Micrococcus luteus and the Marcescens red. So let's see if I can't remind you of how we did that last time. Okay. Remember, we have a low flame. You're going to have not 1, but 2 TSAs. Okay. And you should take your pen, your wax pen, and label one M. luteus and the other one Serratia marcescens, so you know which one's which. You're going to need not 1, but 2 Pasteur pipettes. Okay. So remember, you've already done this once. You did it with E. coli. Now we're going to do it twice because we're rehydrating 2 different organisms. Okay. So you're going to have 2 little vials. They're going to be plastic. Okay. And one is going to be Micrococcus luteus, and the other one is going to be Serratia marcescens. Okay, it's marcescens. And just as before, you're going to take your Pasteur pipette, and we're going to do one and then the other. Okay. So let's say that this is our Micrococcus luteus right here. We're going to go ahead and grab this TSA. We're going to open it. We're going to flame it. We're going to take up 1 milliliter of TSA. There it is. Okay, we're going to flame this guy again. We're going to add that 1 milliliter to your Micrococcus luteus or Serratia marcescens, whichever one you're doing first. You're going to add it in there. You're going to shake it, tap it on your counter. Okay. We're going to resheath this because we're going to need it in a second. We're going to let this sit for about 30 seconds. Okay. After 30 seconds, we're going to take that Pasteur pipette. We're going to suck the entire contents out of that bottle. We're going to take this TSA. We're going to open it, 30-degree angle. We're going to flame it. And then we're going to introduce the entire 1 ml from that bottle. And we're going to flame it. We're going to close it. Okay. This gets thrown away right away. Or let me try it again. I said that incorrectly. This goes into the isopropyl alcohol right away, because you don't want to use this pipette for the other one, because if you do, you're going to cross-contaminate. Okay. Now we're going to mix these on purpose, but you don't want to mix them on purpose here because these are supposed to be pure cultures. Okay. So this is going to go into the isopropyl alcohol so we don't use it again. Okay. This guy, make sure that the cap is only finger tight, because these guys grow better with a little bit of air. Okay. So you're going to do that not once, but twice, because you have to rehydrate the Micrococcus luteus, and then you have to rehydrate the Serratia marcescens. Okay, so we're almost done, guys. What have we done for the second part of this? Well, what we've done is we've rehydrated Micrococcus luteus, and we've rehydrated Serratia marcescens. Now, it's kind of a waiting game. Okay, yeah. Again, you have to take those two tubes and put them in a nice warm place that's safe, that nobody will get to them, and let them grow for 1 or 2 days. Okay, they both have to turn turbid. They both have to turn cloudy, hopefully about the same cloudiness. Okay, so that means that 1 or 2 days later is when we finish this experiment. Or actually, that's not true. 1 or 2 days later is when we continue this experiment. Okay, and that's what I want to talk to you about now. So what we're going to do, we're going to have 2 tubes that are turbid. One is going to be the Micrococcus luteus. The other one is going to be the Serratia marcescens. Okay, and as I told you, we're going to purposely mix them. So we're going to go back to flaming, turning a little Bunsen burner on. There it is. And we're going to take two Pasteur pipettes, and we're going to take an empty culture tube from your kit. Okay, an empty culture tube, and we're going to purposely mix it. So let me get prepared here, because I just realized that I don't have my mask on or my my glove. Okay, so let's say that here's our 2 medias. We're going to need 2 medias, Micrococcus luteus and marcescens red, an empty tube, and 2 Pasteur pipettes. Okay, and what you're going to do is you're going to twist the cap, make sure it's nice and tight, and then you're going to shake it a little bit to end up with a homogeneous solution. Then we're going to uncap it. 1, 2, 3 over the flame. We're going to take some solution out of there. 1, 2, 3 over the flame. Okay. And now I'm going to take my empty. We're going to put 10 drops in there. One, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. We're going to resheath this. Okay. Label it. That way you know that which culture it goes with. And we're going to grab a second one. We're going to grab a second one. And we're going to grab the second culture. Okay. So a second pasture pipette. Here's a second culture. And you go ahead and flame this guy. 1, 2, 3. Take a good amount from there. 1, 2, 3. Grab our culture tube, and we're going to put 10 drops in there. Okay, there it is. Resheath this guy. If you're done, put in 70% isopropyl alcohol. And now, here we have a mixture, okay? So this should be a mixture of Micrococcus luteus and Marcescens red, 10 drops of each, purposely mixed. And now what we're going to do is we're going to try to isolate it, okay? So we're going to take a fresh, clean loop. We're going to take a nutrient agar plate, okay? You're going to take this in here, get an inoculum, just like I am right now. Go out. Take your Petri dish, just like before. Thumb, push the lid with the thumb. It opens it a little bit. See, it opens it a little bit. And then with this finger, we're going to open it a little bit more. And then with this guy, you have to pay attention. This gets a little bit complicated. We're going to go to the far end. We're going to go to this end over here. Put it on there, and we're going to do about 1/4 to 1/3. That's it, no more. 1/4 to 1/3. So I can see where I did that. I'm going to take this guy, and I'm going to put in my 70% isopropyl alcohol. I'm going to grab another one. Okay, this sterile. I'm going to turn this Petri dish, and with this clean one, I'm going to go to the other side again and do it again. About 1/4 to 1/3. I'm going to turn it. I'm going to grab a new loop, and I'm going to do it again. Okay? So here's what we're trying to do. Okay, pretend that, here, let me pause for a second. Okay, so this is what I tell my students to do here, okay? Grab a pen or a pencil, take a piece of paper, draw a circle on it, which is supposed to be your Petri dish. Okay, there it is. And then the pencil or pen is supposed to be your loop, okay? And what you're supposed to do is you're supposed to start on one side, about like that, okay? Now you're going to grab another loop, okay, a clean one, and you're going to turn this, and you're going to put the loop right here on this little corner. Do you see that little corner right there? Let me fold this so you guys don't have to see that. Terrible writing and coffee stand in the back. But there's that. See that little corner? There's a little corner that's being made between the part that I just inoculated right there. See that? At this little corner, I'm going to do it again. Just like so. See that? And then I'm going to turn it, and there's another corner right there. See a little corner? I'm going to put the next clean nichrome loop right there. And these little streaks, they should be visible on your nutrient agar. Okay? So you put the next nichrome here, and you're going to go like this. Okay? Now here you have to be careful. Here's my first streak. Here's my second streak. Here's my third streak. Notice that my third streak went into the second streak, but it didn't go into the, let me try it again. Notice that my third streak went into my second streak, but it did not go into my first streak. Okay? In other words, the last one goes into the one before and nothing else. Because what you're trying to do is you're trying to decrease the cell density. So you go from heavy cells to lighter cells to even lighter cells to even lighter cells. So in the last one, we're going to go right here on this little corner right here, and we're going to do it one more time. And now we're going to use the rest of the media in order to make the cells dilute as possible, okay? And what's going to happen is that here you're going to have a carpet. Here you're going to have a line of cells. Here they're going to start resolving into individual colonies. And over here you'll have well-defined colonies. And that's what you're going for in isolation, is having well-defined colonies on this last streak, okay? So that's what we're going for, and good luck, okay? Thank you.